of it. Now the third step that we are gonna do is to predict the allergic nature of the antigenic protein. That's what our third step. It again comes under the prediction of functional properties. The first property was antigenicity, the next is allergenicity. So here let's again look at two different servers which check the allergenicity. So if an allergic protein is targeted, then what it means is body will suffer from allergic reactions, even anaphylaxis, severe allergy. Okay, so we do not want the protein to be allergic. Okay, so what our intention is, our protein of interest should be an antigen and a non-allergen. Okay, so the first tool that we'll be looking is AlgPred tool that is developed by Dr. GPS Raghava and his team. So go to this particular link and let's start with the process. So let's search in the Google for AlgPred tool. Let's take the second result. Okay, so either you can go to the link or you can search like this. So this is prediction of allergenic proteins and mapping of IgE epitopes. Okay, so here you can give your protein sequence name, which is optional, but let me just give a spike glycoprotein. It was SG that I've mentioned it as a short name. Okay, here what you need to do is paste the protein sequence in plain or standard format. So paste the FASTA sequence. Standard format. So either if it's mentioned as plain format only, then you need to remove the FASTA header. Standard format that we are using is FASTA format. So it's okay if we have the FASTA header. So let's just copy the sequence. I've copied it. Control C, Control B. Okay. Or either you can upload your sequence file. Now if the prediction approach changes as per your research aim. Okay. So here, which means is, this is what I meant, choose the prediction approach. But before that, let's select the sequence format. Amino acid in a single letter code, standard sequence format. So we have used FASTA format, right? So let's just click on to standard sequence format. Now if there are different prediction approaches, mapping of IgE epitopes and PID, then MEME or masked motif, SVM module based on amino acid composition, based on dipeptide, then if you want to do blast search, hybrid approach, but let's just make it as default. Now if your research aim says different, you can choose the different approaches as well. Now let's go to help and see what it says. Okay, so in AlgPred help page, see they have mentioned what are these prediction approaches, IgE epitope. So what it does is, in this approach, the server will search known IgE epitopes. So you just have to read this. Now our default option was SVM module based on amino acid composition, right? So let's read only that. But for you to understand, please read all the information that is given in this pitch. Okay. So here the third result, which was already there as default in the software. So which means that it's generated using amino acid composition of protein sequence of allergens and non-allergens. Here the threshold value that the software uses is -0.4. So at this value, sensitivity and specificity of this method, which means they have done studies and then they have come to a conclusion that its sensitivity is 88.87 and specificity is 81.86, which is pretty good using 5-fold cross validation. Okay, so as you can see, if you use the other method, the threshold value varies and the specificity also varies. So it's again, that's why I mentioned that it completely depends on your research aim. Okay, hybrid approach means by combining all these things. That's what it is. Okay, so we want, our aim is to check if it's an allergen or a non-allergen. So that's why we have moved on to the third or the default section. And then after doing all this, so now we have done everything, right? Submit the sequence. So after that, wait for the results to come, and here the result is. So see the decided threshold, or let me just start with the first. So name of the sequence, length of sequence was 140, and now prediction by this approach. So it's predicted as non-allergen. So the threshold we already know that the software uses is minus 0.4, right? So it's above the threshold. Okay, score is 0.5, which means it's a non-allergen. So positive prediction value is 18.21%, negative prediction value is 71.24, which means it's a non-allergen. Okay, sorry, did I mention that it's above the threshold? It should be below the threshold. If it's above the threshold, it means it's an allergen. So there is a minus here, as you can see. It's minus 0.5 and this is minus 0.4, which means it's the value, it's less than the threshold. So value less than threshold indicates that it's a non-allergen. So if an allergic protein is targeted, so let's say that this is 0.5, which means it's above the threshold, then it says it will be allergen, right? So if it's allergen, then the body will suffer from allergic reactions. So never choose the protein which is an allergen. Okay, so now we know that it's a non-allergen. Now let's go to another server. Okay, let me just show you the slide. Yeah, so let's go to the next server, which is allertop v 2.0. So go to this particular link, search for allertop v 2.0 and look for the, or click on the first result. So when you click on it, you'll get this. So here, paste the sequence. You just have to paste your sequence and remove the FASTA header. Okay. And then click get result. So you can't get the result. So this was what we were looking for. Okay, let me just take it again and show you. So we have clicked the first result, and we have pasted the the protein sequence by removing the FASTA header, and then we have submitted it. Okay, so this is your result. Even here the result says it's a probable non-allergen. Here there is no threshold value which is shown. However, there are chances of getting contradictory results from different servers, like what happened in the antigenic checker, right? The scores were different. Okay, so you need to check if it's a non-allergen and if it's an antigenic. Even though the scores were different, that's okay, but you should not take an antigenic sequence where in one server it says it's an allergen and the other server, or an antigen, and the other server it says it's a non-antigen or a non-allergen. Okay, the result has to be same at the end of the day. Okay, so I just wanted to show you the page of allertop. This is how it should be. Okay, Bioinformatics tool, it's just clearly mentioned that you have to enter a protein sequence here as a plain text. That's why we removed the FASTA header, and we have submitted it by clicking on get the result. Okay, now we know the result. Our next step is to add in the Excel sheet, make another column as allergic score and allergic nature, and the other column as allertop nature. Okay, so I've made other columns: allergenicity prediction, the two softwares, the score, nature. Now copy the score and nature of the allergic result that you have received. It was 0.598, right? So now copy the score, Control C, and then paste it here. And the nature was, it was a non-allergen. Okay, paste the same here. then paste the result of allertop as well, which was again a probable non-allergen. Okay. Similarly, you do all the results that you have got in your other proteins as well. Okay, so we'll go with those proteins which are antigenic but non-allergic in nature. Also select the protein which show the same result when it is checked in any server or software. Okay, so what you need to do is you need to shortlist the proteins which are antigenic and non-allergic in nature, and then go forward with the next step. Okay, so I have made the Excel sheet, I've beautified it and made it in the same format. Now next, let's go with the next step, which is prediction of physiochemical properties of antigenic protein. Okay, so that's what our next step is. So now we have finished with the functional properties. Okay, we have reached till step 3. Next is prediction of physiochemical properties of antigenic protein. Okay,